My name is Jason Vanstone. I'm the research scientist for the Stewardship and Clinical Appropriateness program with the Saskatchewan Health Authority. Today, I'm just going to give sort of a brief overview of the background behind antimicrobial stewardship, what it is, and why the program is important. So in order to do that, we have to start by talking about bacteria. That's really where everything lies here. So if we look at the tree of life, and this is an example of the tree of life, this point right here would be the very first living thing on Earth. So that's where everything starts from, and it branches up from there. Everything in the red, that is the eukaryotes. So that would be all of the animals, everything that runs and jumps and climbs and swims and crawls and slithers across the face of the Earth. That also includes fungi, any of the plants, everything that's green basically that you see, algae, various unicellular eukaryotes, so those are all the protozoans there. So all of that stuff falls just on that one branch of the tree of life. And if you're still lost, that's where people are. We are animals, and that's where we live. This green branch here, these are the archaea. So these are kind of like bacteria, but they're usually referred to as extremophiles. So these are things that live in really crazy environments like deep sea vents, salt lakes, things like that. Environments that are very difficult for most living things to survive in. Which leaves the rest of the tree, the blue pieces here, and these are all bacteria. So you can see, basically from this, that there are a lot of them. So there are many, many different branches along that tree, and each of those represent different types of bacteria. And they've been around for a really long time, which in evolutionary terms it means they're really good at what they do. So if you look at this calendar, clock, whatever you want to call it here, of life on Earth, you can see if you start with the top line, 12 o'clock being present, if you go back about 200,000 years, that's when we have the first humans on Earth. So there's that little orange blip at the top there. The next line over, that's 65 million years ago, that's when the asteroid smashed into the Earth and wiped out all of the dinosaurs. And if you continue to follow that clock all the way around back to that where it says origin of the Earth, so that's 4.6 billion years ago, that's when the Earth started. And in between there, all that yellow stuff, that yellow line, that's the prokaryotes. So this is bacteria for the most part. It's about 3.8 billion years that we've had bacteria on Earth. So they've survived multiple mass extinctions. They've been through all the different iterations of things on Earth, and they've been around for a very long time. So as I said, evolutionarily, it means they're very good at what they do. So a little bit about bacterial cell structure. This is a generalized bacterial cell, and they contain some core things that every living thing needs to survive. So one, you have to be able to protect what's on the inside from what's on the outside. So in the green there, the green layer you can see is the plasma membrane. And that's a cell structure that is common amongst all things living on Earth. So our cells have plasma membranes, plant cells have plasma membranes, as do bacterial cells. Bacterial will also often have a cell wall, so that's the yellow layer there. It's a little bit more of a rigid structure, helps give a little more shape to bacteria, and again, just further protection for keeping the bad stuff out and the good stuff in. And some bacteria will have a capsule, so that's the red piece there. That's just another extra thick layer, helps prevent against desiccation, things like that. So if you're a bacterium living in a puddle and the puddle dries up, it will help your cell from drying out. Another thing common to all living things is the need to store and share information. In living things, this is done with DNA. So bacteria have DNA, just as our cells have DNA. In bacteria, they don't keep it in a sort of separate membrane-bound structure like our nucleus. So here it's just all the DNA sort of one big circular chromosome, all wrapped up on itself like spaghetti, and it's called the nucleolus. So there it is sitting in the middle of the cell. And bacteria also have these extra chromosomal pieces of DNA. These are called plasmids. So they're these little pieces of DNA that are separate from the main chromosome. Oftentimes in bacteria, they will contain genes that will confer antibiotic resistance to the bacteria. So they're very important for bacterial life and for bacterial evolution. Cells also have to be able to grow, so you have these little blue spheres floating around inside the cell. These are called ribosomes. They are essentially little biomachines. They're little cellular machines that will take the information in your DNA and translate it into proteins. So you get, this is how the cells create enzymes and all the different proteins that a cell needs to be able to grow and survive. And as I mentioned, bacteria are very, very diverse. So this is just a smattering of some of the tip of the iceberg in terms of some of the things that can infect humans. Lots of different disease-causing organisms on here that have been important to humans both throughout history and even in current times. And a lot of these that are on there are species which exhibit antibiotic resistance, which is also something important to us in modern medicine. And we'll talk about antibiotic resistance more as we go on here. But first, I want to just highlight a difference between bacteria and viruses. So viruses are essentially DNA or RNA, some sort of nucleic acid, inside a protective protein shell. They're also quite diverse, so you can see here a number of different types of viruses. The flu is there, bacteriophages, which are viruses that infect specifically bacteria. The HIV virus is on there. Tobacco mosaic virus is an example of a virus that infects plants. And of course, Ebola is on there as well. So various different shapes and sizes, but for the most part, the thing they have in common is they are DNA inside a protective shell. And they have a very different life cycle, if you could call it that. So here we have, on the left-hand side, a viral particle coming in and infecting a cell. Maybe this is an influenza virus, and it's coming and infecting one of your cells lining your lungs. And so the virus gets inside, and you can see that protein shell opens up. The viral DNA goes inside our nucleus, and it basically hijacks our cellular machinery as a way of using our cellular machinery to create more of the viral proteins, to create more of the viral DNA, and then that packages itself up and gets to sort of a critical mass, and it will explode out of the cells like little death fireworks, and they go on to infect other cells. We squeeze them out all over our friends and family at school or work or wherever we are. So this highlights a fundamental difference between bacteria and viruses. Viruses, strictly speaking, are not living things. So in order for a virus to make more copies of itself, it has to infect a host and use the host's cell machinery to replicate itself. So as long as there have been humans, there have been bacteria to infect us and viruses to infect us. And this, throughout human history, has been a huge burden on humanity in terms of what people are dying from. So this graph here shows data from the U.S. CDC, and it's comparing the deaths from infectious diseases in 1900 versus, say, 2010. And so in that about 100-year time span, you can see there was a huge change. But what we're looking at is, in 1900, 46% of the deaths were from infectious diseases. So almost half of the people who died, just over 100 years ago, died from either viruses or bacterial infections as your main examples. And you see over that 100-year span that changes drastically, so that by 2010, we see 3% of people who die from infectious diseases. So that's a huge shift in what it is that people are dying from. And a big part of that, obviously throughout that century, there were many medical advances, but a big part of this, because of the infectious diseases, is the discovery and proliferation of different types of antibiotics. In concert with that, if you look at life expectancy, so in 1900, if you were lucky enough to live to your 5th birthday, because childhood mortality was a huge issue back then, but you could expect to be about 45 years old. That was the life expectancy in 1900, it was 45 years. And by 2010, that's up over 80 years. So again, not just a huge change in what people are dying from, but also how long we get to live. And again, there is a lot of modern medical advancements that have come along with that, but certainly one of those factors would be antibiotics. So let's talk about the discovery of antibiotics. So this is Sir Alexander Fleming. He was a physician, scientist working back in the early 1900s in the UK, and he was a bacteriologist, so he studied bacteria, and he went on vacation and came back to his lab after a while and was looking at one of his plates and saw this. So you have these bacteria, this bacterial culture here. That's what the sort of grayish-white streaks are going across the plate. And then there was this fungal colony growing on there. And being a scientist, he thought to himself, isn't that interesting? Because he's scientist. Thank you, yes. And what's interesting, so what he came across was this zone of inhibition, growth inhibition. So there's this area between the fungus and the bacteria, that cleared out circle there, where the bacteria aren't growing. And so what he thought was, well, there must be something from this penicillium fungus that is causing these bacteria to be unable to grow there. And so what we stumbled upon here essentially is this chemical warfare going on between different microorganisms. So these are these creatures, these microorganisms, have to compete for resources in order to survive, just like every other animal on Earth has to. Except these guys don't have claws and teeth and nails to fight each other, so they use chemicals. So penicillium releases what we've termed antibiotics, this chemical that we call penicillin, and it's able to kill bacteria. And in doing so, it prevents those bacteria from being able to grow right up next to the colony and use all of the resources and the food that's available, in this case in the agar plate that they're growing on. So we stumbled across this, and this was sort of the starting off point for the discovery of antibiotics. So penicillin was the first antibiotic, and you can see throughout the middle of the 20th century, there was this explosion of discovery of different types of antibiotics, different classes of antibiotics, as we call them. And they work in different ways to kill different types of bacteria. And as I said, these have been created a huge shift in our ability to fight off and fend off these diseases, these infections. So just to talk quickly about how these things work, so we have our bacterial cell there again, and these different classes of antibiotics, as I mentioned, they work in different ways. So if you have things like beta-lactams or glycopeptides, these work by inhibiting cell wall synthesis. So they prevent the cell from being able to create that cell wall to protect itself. You could also have things like cyclic lipopeptides, which depolarize the plasma membrane, which is fancy science talk for punching holes in that plasma membrane. That creates this ion exchange, which allows for water and ions to flow in and out of the cell, and essentially the cell explodes from the inside, which is a terrible way to die, but who cares because they're bacteria. You could have aminoglycosides, tetracyclines, macrolides, oxazolidinones. These are all different types of antibiotics that inhibit protein synthesis. So if you remember those ribosomes, those little spheres, those little machines that create the proteins for the cell, if you affect those in some way or another and prevent their ability to work properly, you can prevent the cell from being able to build the proteins it needs to survive. You can also inhibit DNA replication, so you can prevent the cell from being able to make and store its DNA. And most of those antibiotics are in the realm of things that we would call bactericidal, so they'll actually kill bacteria, but you can also have drugs that are bacteriostatic. So rather than killing them, they will simply slow them down, put them into a stasis, so to speak, prevent them from growing as quickly. And so things like sulfonamides work in that way, where they'll inhibit enzyme activity. So if you can inhibit different metabolic pathways, for example, you can slow the cell down, have the bacteria grow slower, and that allows your immune system to get a leg up and be able to wipe out the bacteria that are affecting the body. But essentially what this means is antibiotics work by either preventing the ability of the cell to protect itself, to grow, or share and store its DNA. And so just to drive home that point here, that this is why antibiotics don't work on viruses, because as we've seen, viruses don't have any of these cellular structures or machinery. They rely on their host in order to provide these mechanisms for them to replicate. So antibiotics work on bacteria because they have these machines, these cellular machines. They don't work on viruses because they lack these very things. So let's move on to antibiotic resistance. This has become a huge issue in modern medicine, and it is something that is causing our stores of antibiotics to be depleted quickly to the point where we're getting infections in the world, bacteria in the world, which are resistant to nearly all antibiotics, and we're running out of things to treat them with. So antibiotic resistance is essentially a genetic trait. So if you look at your plain old run-of-the-mill bacteria down there, when we throw antibiotics at them, they die. And that's really what we're going for when we're treating people with antibiotics. But if you're a person who has a superbug, like one of these guys, then essentially what it is you have genes that are providing you with some sort of antibiotic resistance. And that's what sets them apart from their weaker cousins. And antibiotic resistance, so just as there are many different ways that antibiotics can affect and kill bacterial cells, there are that many ways that antibiotic resistance can be formed. So, I just sort of color-coded some genes here. So if you have the green gene in the top right there, that maybe creates a drug inactivating enzyme. So it'll create an enzyme in the bacteria that as soon as the antibiotic, which are those little blue spheres there, as soon as the antibiotic gets inside the cell, the drug inactivating enzyme can attack it and basically break down the antibiotic so it doesn't have time to do whatever it's supposed to do. You could also, for example, with the red gene there from the plasmid, have a modified drug target. So let's say that was one of your ribosomes. If you modify the binding site on the ribosome, the antibiotic can no longer bind and prevent the ribosome from working. You could have modified cell wall proteins or drug efflux pump that prevent the antibiotic from getting in or staying inside the cell too long. All kinds of different ways that antibiotic resistance can work. And since this is a genetic factor, we can actually study the genetics and sort of the evolution of this process. And so they did this really cool experiment at Harvard a couple of years ago. And so this is essentially a giant, table-sized agar plate. And what they did is at the ends, they left it so that there was no antibiotic. And then you can see with each sort of section, those vertical sections as you move inwards, they're increasing the concentration of antibiotics. So there's a 1 times concentration, and then the next layer is 10 and 100 and all the way up to 1,000. So that by the time there are bacteria growing in the middle, there is 1,000 times more antibiotic than there is on the outsides. And so what they did is they plated these bacteria, and they let them grow in from the edge, and you can see they basically form a carpet over the top of that agar plate until they get to that edge where the antibiotic is introduced. And there they wait and grow and use up whatever food that they have until a bacteria is able to evolve and develop an antibiotic resistance gene. And then you'll see they start spreading out and moving into the next section, and they'll grow right up into the edge until there's a higher concentration of antibiotic, and so on and so forth. So if we speed this up here, you can see that there are these sort of fan-like outbursts where you get these bacteria that grow and move into the next section. And that is an example of an evolutionary event where you have a bacteria that develops antibiotic resistance, and then all of its progeny that come after it, all the daughter cells that come after it, are able to grow into that next section. And it just continues on into the middle. And if you draw sort of an evolutionary tree over that, you can see all these different events, and you can follow it where, starting at the dark blue, you'll have bacteria whose progeny grow and mutate and get new mutations to the point where they can get all the way into the middle, where you have bacteria that are 1,000 times more resistant to antibiotics than their great-great-great-great-great-great-grandparents all the way back at the beginning. So this is a very cool process to see, and it shows how quickly this process can happen. Obviously, they didn't grow that quickly. There's a time lapse there, but over a matter of days to weeks, you can get these changes occurring in these bacteria, and they grow very quickly to make more of themselves, and they take advantage of their new environment, and you have these population explosions. And in a matter of time, you can get it from bacteria that had no resistance to bacteria that are 1,000 times resistant to an antibiotic. So, as I mentioned, bacteria grow very quickly, so obviously not quite this quickly. Again, there's a time lapse going on here, but if you take E. coli as an example, which is a common enterobacteria that lives in most of our guts, those guys can, at 37 degrees, which is their optimal temperature and just happens to be our body temperature, those guys can replicate every 18 minutes. So you have one bacterial cell that 18 minutes later becomes 2, 18 minutes later those 2 become 4, and so on and so forth, and you get this massive proliferation, and this is why, E. coli is a very common bug when it comes to food poisoning. And so this is why within a matter of hours after a meal that is contaminated, you can be exploding from either or both ends over the toilet. And so, antibiotic resistance. So there's also the ability of bacteria, they're somewhat promiscuous with their DNA. So there are multiple different ways that bacteria can even share DNA. So here is an example called bacterial transformation. And you will have on the left the donor cell, so this would be a cell growing in amongst a colony of bacteria that dies, and the little red piece there, the little red squiggle, is the antibiotic resistance gene. And so when that bacterium dies and it kind of breaks apart, that DNA gets released into the environment, and other bacteria can actually just take up that naked DNA without having to go through any sort of specialized process to evolve it on their own. So if you have uptake of this antibiotic resistance gene, you now have the recipient cell, which acquires antibiotic resistance without having to go through all the sort of time and trial and error to evolve the resistance on its own. You can also have bacterial transduction. So I spoke earlier about bacteriophages, these viruses that specifically infect bacteria. And so similarly, when they're repackaging up inside the cell, sometimes they will accidentally take a piece of the bacterial genome along with them. And if that happens to be an antibiotic resistance gene, then when they're injecting their DNA into the next cell nearby to infect that one, they can pass along those antibiotic resistance genes. And then there's also bacterial conjugation, which is as close as you can get to bacterial sex. And so what we have here is one bacterium that creates this sex pilus, which reaches out and attaches to another bacterial cell. And through this tube connection, they're able to transfer DNA. So they can actually transfer these plasmids, those little circular extra chromosomal pieces of DNA. And again, you can see on this plasmid it's got some antibiotic resistance genes. So all of these are different ways that bacteria can share DNA without having to go through the sort of trial and error of evolving and then reproducing to share those beneficial mutations. And so that spells bad things for people. These bugs grow fast, and they can share DNA, and they can not only share DNA amongst members of the same species, but even in some examples of cross-species sharing. So you could get different bacteria living in a pond that's contaminated with sewage waste that are able to share antibiotic resistance genes across multiple different species that could then go back to infect humans or other animals for that matter. So, antibiotic resistance, how does this work in people? So, an important message to get out to the public is that it's not people who are resistant to antibiotics, it's the bacteria that are living inside of us. And so if you look inside of people, that's what we look like in our small intestines. And we would have these bacteria growing and living inside of us. Most often they're good bacteria, so they're not harming us. They're aiding with digestion. They help us to get all the nutrients we can out of our food. And sometimes some people might be unlucky and you've got some bacteria that are resistant. So you can see that one has, say, a plasmid with some antibiotic resistance genes. And maybe in one of the ways that we've talked about it, it shares its DNA with other bacteria or other species within you. But for the most part, you've got more good bugs than bad bugs. And because they have these extra pieces of DNA, it takes them a little longer to grow and divide and multiply. And so they can't grow as quickly, and so they're outcompeted for resources by the good bacteria that are growing inside of you. The problem comes when you take an antibiotic, particularly if you don't require an antibiotic. It goes down into your stomach, it dissolves, and it spreads throughout your entire system. It gets into your bloodstream, and it goes throughout your body. And it will kill anything that is not resistant to the antibiotic. And so you're left with this situation, and when the problem occurs when you're done your course of antibiotics, because now this is what's left inside of you potentially. All you've got are these bugs who are resistant to antibiotics, and they no longer have to compete for resources against all those other guys. And so now you can become colonized potentially with antibiotic-resistant bacteria, and you've got a potential now for your next infection to be perhaps an antibiotic-resistant one, and then you can no longer use sort of a penicillin or something like that. You've got to step it up to a higher impact antibiotic, something that has a broader effect on different types of bacteria or might have worse side effects, something like that. So I like to use this to sort of illustrate why it's important to only take antibiotics when necessary. And we know that particularly amongst community prescribers, there is a lot of prescribing of antibiotics for upper respiratory tract infection, and the vast majority of these are viral in nature. So we're giving people antibiotics for viral infections, which we know is not going to help them because antibiotics do not work on viruses. And so instead, we're just giving these people antibiotics, and it's not even doing anything to help their illness, to help get rid of their infection. So when you're taking these antibiotics unnecessarily, you're really then only exposing yourself to side effects of these medications, and then, of course, the potential for developing antibiotic resistance. So another problem for humanity in terms of this antibiotic resistance crisis is a lack of new antibiotics. So I showed this before. We had this explosion of discovery of different types of antibiotics throughout the middle of the 20th century. And what's interesting is if you extend this timeline, you can see that throughout sort of the 1980s and 90s, we hit this development void where really in the past 30 years, we haven't sort of discovered any new classes of antibiotics. We've made improvements, minor improvements to some of the antibiotics that we've had to make them maybe more broad spectrum or help them fight off antibiotic resistance in their own way somewhat, but we really haven't discovered any new antibiotics, new ways of killing off bacteria. And part of this is because of the way the drug development process is set up. So when we talk about pharmaceutical companies creating new drugs, for example, if a pharmaceutical company was to set out to create or develop a new antibiotic, really they start with this drug discovery thing. So they scan through about 10 to 15,000 different compounds, and then over a period of sort of 3 to 6 years, they'll maybe weed that down to sort of maybe 5 compounds or something that will indicate like they're probably going to work for the problem that you're looking at and are considered safe to use in human volunteers. That moves from your preclinical work after like about 6 years to the clinical trials. They have to go through several phases. This could take about 6 to 7 years on its own, and then maybe 1 or 2 years after that, you might get one FDA-approved drug. So you're going from these huge libraries of over 10,000 compounds over 10 years and like $1 billion just to get one FDA-approved drug. And so when you think about that from this pharmaceutical company perspective, there's not a huge payoff for the investment that they're making in developing these drugs, especially considering the fact that when they do create a new antibiotic, we simply say thank you and we place it on the shelf for 10 years because we want to ensure that when all the rest of the drugs stop working, we'll have something to go for. So by the time we start using that antibiotic, it's definitely gone beyond any patents, and so other companies can make generic versions of the drug, and the price of the drug drops down. So there's a big problem there, and governments and industry are working together on this to try and find ways to entice pharmaceutical companies to get back into this and spend the time and effort on creating new antibiotics so that our pipeline of new antibiotics isn't drying out so rapidly. So again, antibiotic resistance here it's a genetic thing, and it's something that is going to happen and has happened with every antibiotic that we have discovered to date and or created in a lab to date. And so on the left here, that's what this graph is showing. On the left is when an antibiotic was first introduced, and on the right is when we first identified the antibiotic resistance. So you can see with penicillin at the beginning there, first started being used around World War II. Within about 20 years, we were identifying penicillin-resistant pneumococcal species. And, you look down the line there, and it's about 10, 15, 20 years for most drugs. Levofloxacin is one of my favorite examples because as soon as we started using that drug back in 1996, we already knew that there were levofloxacin-resistant bacteria out there. So, in some cases with newer antibiotics that we've found or created, we don't even have that sort of luxury of that 10 to 15 year timeline to use that antibiotic without seeing resistance. And we've seen these skyrocketing rates of antibiotic resistance. This is data from the U.S., but it's somewhat similar, although maybe not as sharp of an increase in Canada. But you've got things like MRSA, which is our methicillin-resistant Staphylococcus aureus, VRE, which is your vancomycin-resistant enterococci. There's fluoroquinolone resistance there. But you see these just huge, sharply rising rates of antibiotic resistance throughout the 80s and 90s. And it is our overuse and improper use of these antibiotics that drives this. The more we use these antibiotics, the more resistance we see. And in Canada, so that was data from the U.S. In Canada, it's we're no better here in terms of resistance rates for a lot of things. This here is percent of gonorrhea isolates demonstrating resistance or decreased susceptibility to various antibiotics. So it's data from the Public Health Agency of Canada. And you can see over the years, so from 2011 on the left through 2015 on the right, we see these rising rates of resistance to various types of antibiotics amongst gonorrhea. Interesting fact, it does happen to occur most often in the baby boomer population. There's all kinds of interesting social pieces that go along with that. But essentially what you're looking at here, tetracycline resistance, for example, has gone from 30% resistance to almost 60% resistance in that sort of 5-year span. And so that's quite alarming, and other things are raising too. So we see ciprofloxacin resistance here. And we're getting multi-drug resistant isolates of gonorrhea that are showing up as well. So that's a concerning trend. So I mentioned methicillin-resistant Staph aureus. So we have the infection rate per 10,000 patient days. You have all MRSA infections at the top here, and you can see it's a ever so gradually increasing line. And then your bloodstream infections along the bottom here. So these are infections with methicillin-resistant Staph aureus. And if we look at that by region too, we can see that in the West, we are at the top of the ladder when it comes to the highest, having some of the highest rates of incidence throughout Canada. So, and this is, as I was saying, this is data that shows the more antibiotics we use, the more antibiotic-resistant bacteria we see. So in the winter here, in the blue, we've got the aminopenicillin prescriptions, and in the brown, we see E. coli resistance to Apiso. So in the winter months, the more antibiotics we use, you can see there's about a 1-month lag, but the more antibiotic resistance we see in E. coli. And as we use fewer antibiotics, we see less resistance in the E. coli. But as we start to use more, when we get into the winter time again and everyone goes back to school and starts coughing and sneezing all over each other, we see higher rates of use and higher rates of resistance in E. coli. And in Saskatchewan, again, data from PHAC here, we are the second highest user of community-prescribed antimicrobials. So looking at all the different provinces and territories across Canada, we are the second highest user of those community-prescribed antibiotics. So things that you would get at your local community pharmacy, prescriptions made by family physicians, emergency outpatients, those types of things. So it's not a silver medal that we want to have, and we certainly have room to improve our prescribing practices here in Saskatchewan. So what can we do about this? First of all, not requesting antibiotics when you're diagnosed with viral illness. So physicians are people, they're humans, and they will succumb to pressure. They want to do right by their patients, but we certainly don't want to be forcing their hand and trying to make them prescribe you an antibiotic when you clearly have a viral illness if you've been diagnosed as such. Getting your flu shot, so preventing illness means fewer trips to the doctor and less chance of actually getting an unnecessary antibiotic. And of course, practicing proper hand hygiene, ensuring that you're washing your hands is another great way of protecting yourself from getting sick in the first place. So if we don't do anything about this, there was a review on antimicrobial resistance in 2014 released in the UK, and they, through some very fancy number crunching, decided that if we do nothing about this, then by the year 2050, we could see more than 10 million deaths a year from antimicrobial resistance. And that's certainly not a place we want to get to. And just to put that in context, that would be more deaths from antibiotic-resistant infections than cancer, diabetes, and so on and so forth down the list there. So when you think about that, compared to that graph that I showed at the beginning, where we saw the change from 1900 to 2000 in what people are dying from, this actually indicates that there might be a change in that graph, and it'll start going back the other way to the point where we're almost returning back to the dark ages of medicine, so to speak, where we've got antibiotic-resistant infections that once again become a real threat to people and can affect things like how long people get to live. So with that, that brings us to the end of the talk. Thank you very much for listening.